Imagine a world without hunger, disease, or even death. This can be achieved with Crisper Cas-9, or clustered regularly interspaced short prodromic repeats. But what is CRISPR? Back when bacteria ruled the earth, CRISPR was used like an early immune system. When a virus would enter a bacteria, it would insert its own DNA into the bacteria's DNA so that it could spread and multiply and then eventually kill off the host. If the bacteria were to survive this, it would save the virus's DNA in a place known as CRISPR. If the virus were to ever attack again, the bacteria would create an RNA copy of the saved DNA. The RNA is then delivered into a protein known as Cas9. From here, Cas9 scans the bacteria's DNA, and if it finds the virus's DNA, it cuts it out of the DNA strand, rendering the virus useless and eventually die. But why is this important? This is because scientists have found a way to program this system. Here's how it's done. Scientists go into an organism's DNA and find some mistake or change that needs to be made. They program the genes they want into the Cas9 protein. Then the Cas9 travels until it finds what needs to be altered. Guide DNA is also injected into the cell to help guide Cas9 to the right location so that it cuts into the correct spot. Once the strand is found, Cas9 cuts it out of the DNA sequence. Immediately, the cell realizes that the DNA is broken and begins to repair itself. The guide RNA steps in and becomes bonded, completing the DNA strand. Then the cell continues on and spreads its new DNA. This allows scientists to completely change the structure of an organism. Imagine the possibilities. If someone had cancer, all scientists would have to do is inject CRISPR into the body, and then the cancer would be completely cut out of their DNA. You could even make yourself immune to cancer altogether. Besides cancer, other genetic defects like autism or Alzheimer's could be eradicated from your DNA forever. We would also be able to transform agriculture. We could modify plants to be resistant to insects or other hazards such as toxins. We can make them need less water and to produce more crop. World hunger would be a thing of the past. The technology could also help us colonize the universe. We could edit the genes of humans to be better equipped to thrive in new environments. We can make them stronger, taller, live longer, or even breathe underwater. There are so many possibilities, but there is a price to pay. At our current understanding of genetics, we do not know enough about the human genome to ensure pinpoint accuracy. We simply do not have enough information to know exactly how our genes work and interact with one another. Errors are still happening all the time, and we don't really know why. The results could be catastrophic. Imagine trying to change someone's eye color, but accidentally make their heart unable to pump blood. This could destroy the human race if we are not careful. By changing humans' DNA, we could change the gene pool entirely. The gene pool would be less diverse, as most people have a unified standard of what healthy and beauty looks like. The diversity of the human race would become non-existent. One disease could kill off humanity in an instant because we all share nearly the same DNA. Imagine if countries like North Korea with this technology, they would be able to create super soldiers that would be unnaturally strong, intelligent, and unstoppable. Or what if they designed a bioweapon targeted at a specific group of people with similar DNA? This would be horrendous. With all the cons, it would be easy to say that we should give up on this technology, but that would be a mistake. There's so much good that can come from it. Giving up on this would be like giving up on medicine. Dangerous in the past, but beneficial in the future. There's so much good that can come from this technology. As long as we keep in mind what could happen, we can continue to grow as a species and one day conquer our own biology.